of Protein Design. And there I studied at the Institute of Protein Design with my advisor, David Baker. So my specific research there was specifically applying all the advances within machine learning and seeing them, we can apply them to the protein design problem. And today I'll actually be talking about one of the stories I worked on while I was there. And now I am currently in New York at Prescient Design. And Prescient Design is a company that tries and utilizes the advances in machine learning to expedite the drug design process, both at the discovery stage and at the lead optimization stage. And so the general outline of the talk is first I'll go over the method, which would be part one, and then I'll go over the applications. So let's just go ahead and get into it. Also, feel free to stop me while I'm talking to ask questions. That would make sure that I'm not losing people, and it'll be a more fun talk that way. So some preliminaries of what exactly protein design is. So first, let's define what a protein. So a protein is just a long polypeptide chain composed of functional groups called amino acids, and these functional groups have certain biochemical properties, to which point when they interact with each other, it gives rise to a structure. And this structure usually confers that protein with some functionality as it relates to whatever utility it has. And so a lot of time this functionality is both dependent on sequence and structure, both either or. So for example, in the case of a structure dependent functionality is in the case where you have function defined by specific small functional motifs. So for example, if you have an enzyme, that enzyme has an active site that catalyzes a given chemical reaction. And so that active site would be the structural dependent functional motif within that protein. But in addition, say you have a protein that binds another protein, there we would define the functional motif, the structural functional motif, as being the interface residues interacting with that said protein's partner. So those are examples of structure dependent function, but you also have sequence dependent function. So in the case you have a functional motif that's entirely sequence dependent. In fact, these sequence motif, and oftentimes if it does take on a structure, that means it usually loses its functionality. So for instance, you could have a cleavage site, and that would be the functional motif there. And then some functions are both dependent on sequence and structure. And so, for instance, if you have a protein that adopts specific states dependent on some environmental stimuli, that is one that's both dependent on the interplay between sequence and structure. And so if you really want to be able to develop a generative model for protein design, you should be able to tackle all these three problems. And so that's exactly what we sought out to do when we developed ProteinGenerator. But then if we take a step back and look at how the typical protein design process has been undergone, it's a very step-by-step process. So first you define some sort of function or design objective that you want. You then figure out what method that gives you a structure that you think harbors that structural hypothesis for that given function. And now this largely hasn't changed. And previously we had, in more classical protein design methods, we had things like Rosetta, fragment assembly, and now more recently, we have more deep learning-based methods like hallucination and diffusion. But then once you have this structure, you then need to go ahead and find a sequence that matches that structure. This is what we call sequence design. Now, again, this hasn't changed largely. So previously people would use Rosetta, now people use deep learning-based methods as well. And then from there, now you actually get your protein, which you can get. You can now go ahead and go to the lab, synthesize and test whether it works. Now, some methods, so specifically language models, they go ahead and hop from the function directly to the sequence. Now, where our method differs is we go ahead and define the given function objective that we want, but we go ahead and concurrently give you both the structure and the sequence. And we think this is generally how the field will go as we move forward. Now, given that we have defined what our objective is and what we want our generative model to be, then the next question is what architecture is amendable for us to actually go ahead and start making this. And so ideally you want a model, or sorry, an architecture that can both take in sequence and structure and both output sequence and structure. So being able to input sequence and structure means we can condition on that. And being able to output both sequence and structure means we can generate both concurrently. Now, structure prediction networks are particularly very good at this. And so that's what we actually sought out to do. We use RoseTTAFold 2, a state-of-the-art structure prediction network, as our architecture. Now, previously people have done structure diffusion with RoseTTAFold, but here we're doing sequence diffusion. And now I'll go ahead and give you some specific reasons why we think it's beneficial to be working within sequence space. So for one, sequence data is easier to collect. So that is, if you're going to run experiments, it'll be much faster for you to get sequence to activity data rather than structure to activity data by virtue of it just being way more difficult to go ahead and solve a structure. Now, because you have this data set, that means you can go ahead and train a classifier to predict the activity just entirely based on a sequence. And through what people have previously developed within the diffusion literature, you can actually use these classifiers to go ahead and guide you to some region within generation space. So for instance, if we take these classifiers and guide our generation process, we can go ahead and get sequences with higher activity by virtue of the data collected in the lab previously. Now, this is a really, really, really important thing. Specifically in the cases where it's more difficult to define a function. So this gives us an avenue to kind of put data onto the problem. And then another super important thing, especially as people try and use protein design to actually make therapeutics, is it's not enough that your protein has a given functionality. It has to have specific developability properties. That is, say for instance that you're trying to design a drug and you have a protein that binds onto a target. It's not enough to just bind. You also want to make sure that your protein does not induce an immune response. And so you want to be able to guide for the sequence-specific properties that do not inherently arise from structure, but mostly arises from the actual sequence of the given protein. And another important thing is, say you're actually trying to design a therapeutic, you need to be able to you need to be able to synthesize these proteins at scale. And so you need a protein that's easily synthesizable and has certain properties such that it doesn't easily aggregate. And so you can guide for these specific properties if your generation process is within sequence space. But in addition, as I mentioned earlier in the talk, structure specifically arises from sequence. So even though we're only manipulating sequence specific properties, these properties actually gives rise to specific structure attributes. So we're still able to both mend sequence and structure by only interacting with it within the sequence space. Okay, so now the next question is, how do we actually represent sequence such the model understands it? And so previously, the literature within diffusion has worked very heavily within continuous space. And so we wanted to be able to leverage all of that literature, specifically in the case of classifier-based guidance, which has been very well defined within continuous space. We wanted to be able to utilize all that machinery people have developed for our sequence-specific properties. And a simple way to do that is we take a sequence and then we go ahead and represent it as a one-hot tensor. And so when we actually go about doing the corruption process within diffusion, it's as easy as adding Gaussian noise onto this one-hot tensor as if it was an image. And here's an example. So for some regions where it's unclear clear what the specific amino acid is, we can go ahead and see if we plot the sequence logo from this one hot representation to an actual sequence. We see if in regions where it's unsure, there's multiple amino acids that can be there, but then in regions which it is more sure, there's only the density is mostly towards one amino acid. And then we can actually go ahead and specifically tell the model that we only want a specific sequence at this position. We don't want it to change. So we can also directly modify the one hot tensor such that only that given amino acid is present at that given position. So here in this case, we have the methionine. We only want the model to put the methionine. And so this representation, while very simple, also very powerful. Now, previously, people have also done very similar formulation. Oh, sorry, go on.
I wanted to distract you a bit about the denoising process in the diffusion step. Yes.
Mm-hmm.
When you are adding in noise, right?
Mm-hmm.
I'm just curious. What is this process like? Because from an image point of view, if we are adding noise to images, we can just add in maybe just random pixels that are not supposed to be there, maybe. So what is the process like here of denoising in at this stage of adding
Yeah.
noise? Sorry.
So when we represent something as a one-hot tensor, a given index would be, so this is an example. If you look at this specific index, at this position, everything would be 0 up until the amino acid that you want. So in this case, this specific row would correspond to methionine. So this would be a 1, and then everything else would be a 0. And so essentially how we do the noising process is we go ahead and have a random number generator that samples a Gaussian, and we specifically add those random numbers to all logits within this within the tensor. And so it's the exact same process that you would do within images, except just imagine. I mean, you don't even have to imagine. Just look at this and assume it's an image, and that's exactly what we noise it as if it's an image.
Thank you. That's great.
Yeah, and then again, just encouraging folks just to feel free to ask questions throughout the entire process. So that way I don't feel like I'm talking to my computer.
No.
Oh yeah, okay. So the formulation I just laid out, there was concurrent work that actually has a very similar formulation from Geoffrey Hinton's group, where here they call it analog bits. Where instead of representing it as a one hot, they represent it as in a bit formulation. But likewise, you could also, in fact, they have a section where you can say if your codebook size is small enough, then a one hot representation should be sufficient. And in this case, our codebook size is 20 amino acids. And so naturally, we can do one hot easily. But then another cool thing they actually did in this paper is they introduced this concept of self-conditioning. And so whereas in the typical diffusion process, in order to get to your next XT step, you're conditioning on the previous noise step, but then within this concept of self-conditioning, you're also inputting the model's X naught prediction in addition to the noised XT. And so what we've actually found, like the authors did in this, is this actually stabilized training and helped us converge faster by quite a bit. And we believe the reasoning is there's a lot of information when the model is actually predicting X naught that gets lost if we throw it away. But if we keep it and use it as conditioning for the next time step, it's actually super beneficial.
Rinsa, please, I have a question.
Uh-huh.
The previous slide, you talked about generating discrete data using diffusion models. So is there a possibility of coming up with continuous data using the same methodology?
Can you ask that question again?
In the previous slide, you talked about
Is it this slide, this specific slide?
wait a minute.
Or, sorry, this one. Okay.
Yes. Generating this discrete data using diffusion models with self-conditioning. So I have a question. When we are dealing with discrete data, usually we compare it with in a continuous context as well. So is there a possibility of you having any kind of research in this area in relation discrete and continuous?
Okay, so I'm gonna rephrase your question because I don't think I quite understand it. So are you asking if it's possible to use this formulation of discrete data generation in the continuous
Context.
in the continuous context?
Yeah.
I would say that we are doing the opposite. We're using how people do normal continuous generation in the discrete context. And so the answer is yes, because I guess that's already what people are doing, if that makes
Okay.
sense.
Oh, thank you. Thank you.
Uh-huh. Okay, so okay, let's go ahead and start putting all these things together. So remember, we're using a structure prediction network as the we're using the structure prediction network as our architecture, and the structure prediction network is both giving sequence and structure, and we are so we're both giving sequence and structure, but our diffusion process is within sequence space, and we apply both losses both at the sequence and at the structural level. And so when we put it all together, we see that we can start from a completely noise sequence. And then we task the model with denoising, giving you both the denoise sequence, but then also the corresponding structure. And so in the beginning, we see that when it's unclear what the sequence is, the structure is also correspondingly jumbled. But then if we continuously denoise, we see that both the sequence and structure are becoming more coherent, and all the way until we reach the last time step where the sequence and structure have been settled, and then that is our design protein. And then one thing to note is how we're actually doing this is we have the model predict X naught, and that is the denoise thing at every
Sydney.
specific. Yes.
About this slide and the previous slide, but let's go to the previous slide. Yes, this one. I'm curious about the X naught that seems to come out of the step when we add in, is it noise, xt plus the delta?
Yeah.
And then we obtain the X naught with
Are you talking about A? You're talking about A right now?
yes.
Yes.
Yes,
Okay.
yes.
Yeah.
And
So
then
the
the process.
model. Yeah, so I guess to kind of enumerate what exactly is happening here. So the model takes in xt, and then it denoises it to x naught, and then we go ahead and noise it back to xt, and then we give this to the model, and it predicts x naught. So that's typically how the normal diffusion process works. But then within self-conditioning, we have the model take in xt, and it produces x naught, and we go ahead and denoise it to xt. But for the next model iteration, it both gets xt and x naught to give you the new x naught here. And so the self-conditioning part is the addition of this specific input to the model.
Okay, that's great. Quite brilliant. Okay, now it makes it clear because I wanted to ask about the standard reverse diffusion step, and on the next slide. Yes, and on this because I was like, since we want X naught, which is the denoised sort of sequence,
Yeah.
and then
Sorry, go on.
on the previous slide, I was seeing we are predicting at every step. I thought we are predicting at every step X naught with a tilde. So I was like, what's the connection between? But now you've clarified the two steps, and thank you.
Yeah. Yeah. And then I guess here I'm also not showing what the model is outputting. I'm showing what the model gets at each specific time point. So it gets this,
Okay.
and it gets this. And then only here when we zoom in, am I actually showing what the model is doing. So in this case, it gets xt. The model takes it in and it predicts x naught. But in addition, we're also predicting the structure. So you're both getting the sequence and the structure. But then we can actually do quite a bit more. We can also go ahead and condition on structural attributes directly at this stage. So in this case, we can give it explicit XYZ coordinates if we're, say, trying to do motif scaffolding. But then we can also do this sequence guidance in this step where we have our outputted x naught. We go ahead and add in gradients from a sequence activity classifier. And then this biases x naught to some region within sequence space with some properties that we care for. And then we go ahead and scale and then add in noise to get our xt minus 1 to be input to the next thing. But then note that what we're not showing here is self-conditioning within this, but self-conditioning would just happen by just concatenating x naught to this xt minus 1. So we take xt minus 1 and x naught and input that to the next step in the model. And then here's just an overall schematic of what we're doing. And so in protein sequence, and so this is the natural world, protein sequence is discrete. And so it's a single tensor. And then here we put it in continuous space by making it into a one hot tensor. And so now it's size L by 20. And then we go ahead and noise it to give us ZT. And then this ZT gets input into the model. And then we condition on certain aspects as well, both at the sequence space in green here and also structure space in red here. That gets passed on to RoseTTAFold. And then that goes ahead and gives us our predicted Z naught. And then here to get back to discrete space, we just argmax, and then we get our predicted X naught. And then for structure space, we don't need to do anything as soon as we're, because we work in structure space directly, so we get Y naught, and then that's overall what we're doing here. And then another important parameter with this in this entire process is how we actually go ahead about noising. When you're working within discrete data, you actually need to give a higher amount of noise to go ahead and corrupt the signal that you would typically do within continuous diffusion space, whereas people there within that space use a cosine noising schedule. Here we found that using a square root noising schedule is actually super important. And the square root schedule is right here, where you see in these lower time steps are actually have a substantial more amount of noise than you typically have, say, if you're using cosine. And so we actually found this super important to actually make sure that we're doing diffusion properly.
A question,
then, yes.
Yeah. Is there any assumptions that are made when you are doing the noise scheduling?
It's a fixed schedule. So the assumption would be that the schedule is the most optimal, or I might not be understanding. I guess what yeah.
Because if you talk of linearity, we assume normality. So is there any did you look at any assumptions for normality in this context?
Normality with respect to what?
Because I've seen you talked of linearity. So we assume linearity when there is normality.
Oh, so I guess, yeah. So I guess here, this schedule corresponds to how much noise. And so yeah, and so essentially the amount of noise you can see within the y-axis. So 0 would be no noise, and then 1 would be full noise. And so this is essentially what is the scaling we're using for the noising we're adding. So if the scaling is small, then there'll be no noise. And then this is the literal function actually displayed of that schedule.
Okay, thank you.
And then there's other training details we incorporated. So specifically the standard diffusion tasks were both noising everything. So that's 40% of the time. And then the structure prediction task where we are noising when we're not noising the sequence at all. And then just tasking the model with predicting the structure, that it goes about 30% of the time. And then the backwind design task where we give the full structure, but give no sequence information, that's about 30% of the time. And then we do different masking ratios for in the case where we're doing motif scaffolding, where it's a contiguous span of residues we want to scaffold, or active site scaffolding, where it's a small number of residues that we want to mask. And so this was in the so this was the masking schedule we did for these motif scaffolding problems. And then we also condition on secondary structure about 25% of the time, and mask to 0 to 90% uniformly when provided. And so this just lets us tackle numerous tasks with the model. And one thing we actually found super useful in terms of getting high quality outputs from the model is actually this backbone design task, because we think it helps the model better understand sequence to structure relationships. And then in more detail about the type of information that's been given to all tracks of the model. So RoseTTAFold is composed of 3 primary tracks, a 1D track that deals with primarily sequence information, a 2D track that deals with pairwise information, and a 3D track that deals with explicit XYZ coordinates. And so for the 1D tracks, here we provide the sequence, DSSP, masking, and the time set information to that. Whereas with the 2D track and 3D track, that's primarily structure, whether that may be in the pairwise relation form or the explicit structure form for the 3rd track. And then this is in general, the pseudocode. I won't walk through all of it, but one thing, but one thing to note is that we apply 3 sets of losses, specifically a CC loss on the sequence avenue, a KL reconstruction loss, where we essentially compare the distribution between the predicted X naught and X naught, and then make sure that those are okay. We actually found this was important for getting high quality outputs, and this was work suggested by one of our collaborators. And then structural losses to give you and then structural losses to make sure that we have coherent structures each time. And then this is our sampling procedure. And one thing to note is how we're deferring from typical diffusion literature is usually people interpolate from X naught to XT. Whereas we found that kind of gave us some mode locking behavior. And so we go ahead and noise to XT without conditioning on the prior input to XT. And we actually found that to perform empirically better. And here we're actually plotting whether we do the typical typical interpolation found in normal literature, and that's what we have in the pink here. And whereas if we just do our version where we just noise the XT without any information about the input XT, and we see we do substantially better within predicted AlphaFold 2 PLDT, which is essentially the model's confidence in the outputted structures. And you see we're significantly higher in all 3 length cases. And then if we go ahead and look at the difference between the root mean square deviation between the design and the predicted structure, we see that it's substantially, yes.
Sorry, but can you explain a little bit where interference is coming from? I'm kind of confused here.
Oh, so inference is when we're actually using the model to do sampling. And so there's a specific...
No, I'm asking about interference. Where is it coming from? Like interference. Oh, sorry. I thought it was interference. That's why I was wondering. It's okay. Sorry.
Oh, okay, good.
Yeah, you can proceed.
Okay, good. Yeah. So essentially, this is different from what other folks do, but we found empirically that this is what worked best for us. And then if we start comparing the actual, yes.
Before you go, yes. So you say that you use the model to estimate the sampling, right?
Yeah, we use the model for sampling.
I'm confused a little bit. So do you have the like you estimate the population for it, and it helps estimate the sample, or it does estimate the population and the sample itself?
So in this case, we task the model with generating 100 structures. And then we take those 100 structures with their sequences and we run them through, we take those sequences and we run them through AlphaFold. And then here we compare how well the AlphaFold predictions of the sequence correspond to the actual structure.
I'm sorry to ask a lot of questions, please.
Uh-huh.
Which criteria do you use to know this? Like there is a significant. Do you use like a p-values or something in that context?
Oh, I understand now. I think I think this was a p-value test, less than 0.001 or something. Actually, I don't
Okay.
remember the exact number.
Okay. So I ask a lot of questions because my background is biostatistics. So when it comes to sampling, that is
Yeah,
a lot of questions there. You
disease
can go.
field.
Yeah, thank you.
Yeah. And then yeah, and then here we're just comparing the outputted distributions from the our model where we generate I think it was 100 proteins in this case, and then 100 proteins of a similar size from natural proteins. And here we're looking at the amino acid distribution differences between those. And one thing that's notable is that our model tends to like using glutamate specifically in high frequency, and also leucine and isoleucine, and also even arginine. But then the model seems to shy away from these more rare amino acids such as cysteine, sorry, such as cysteine, histidine, and tryptophan. Oh, and even tyrosine. And so it's interesting that the model is doing this, and unclear why. But then if we go ahead and actually compare against another protein design model, specifically here, protein MPNN, we see that we seem to be following the same patterns that protein MPNN is using as well. And it might be that it's just it's maybe it's in essence it's some form of mode. I don't want to use the word mode collapse, but it seems like it's picking up on things that are more likely given the structural context. And then if we go ahead and actually compare protein MPNN to our model in terms of if you take natural proteins and then you redesign them with either ProteinGenerator or MPNN, and then you predict those sequences with AlphaFold, can you see that AlphaFold predicts them more confidently or not more confidently? So in this case, if we look at AlphaFold predictions of the native proteins, and again, this is single sequence prediction, we see that the LGC tends to be on the lower end, but for both our model and MPNN, we see that we shifted substantially to the right here. But it seems more so that MPNN is still better at giving you giving you higher PLDT values. So there's still room to grow, but this was a good promising result to see. And then here we're comparing against to another generative model, specifically RF diffusion, but this one primarily deals within structural space. And then if we compare again with this AlphaFold metric, we see that we're comparable, but the model does do quite a bit better within this longer length regions. And we're thinking it's just harder to model long-range sequence dependencies, and it's pretty powerful if you're also doing diffusion within structural space, because those long-range sequence dependencies are apparent. The model it has better reasoning that they're not actually long-range if they're close in tertiary structure. And then here again, here's another benchmark. In this case, we're doing motif scaffolding. That is, you have a functional motif. Can you scaffold it, and does AlphaFold predict it accurately? And here we're seeing we do get some successes. We're sort of we're not particularly strong in this. But then one thing to note is we're using the same model in both these cases. Whereas with RF diffusion, they have specific trained models both for unconditional generation and motif scaffolding, and so in essence, they were trained for both those tasks. Whereas the model we're showing here and all these three benchmarks is the same model, same weights, and so it's nice seeing that it has versatility in all these objectives. And then here we're comparing to just sequence-based models. And so here, interesting enough, the metric we're using here is ESM perplexity. That is, if we take a language model, specifically a protein language model, predict specifically ESM, do we see that the model thinks that the sequence outputted, are they natural or are they not? And so if we take a random baseline, so we see ESM perplexity is high, and then we go ahead and take another, another sequence diffusion model, EvoDiff, it actually does pretty poorly. But then if we compare ProteinGenerator with actual natural proteins, we see that we both perform substantially well within this benchmark. So that's really nice to see. And then if we go ahead and take EvoDiff and actually compare it on AF2 prediction of said AF2 predictions of said sequences, we see again we perform better even in this sense. This was really cool to see because EvoDiff is a massive model on the scale of 1 billion parameters, whereas our model is like 20 million. And so that's cool to see. And we think that this kind of comes down to our model has an intrinsic understanding of that sequence to structure relationship. So it really allows it to excel in this case. Okay. That was the method section. And now we're actually going to start going into some experiments to actually verify that our model can actually generate things of use. Okay. It seems I'm also running out of time, and so I might go through some sections.
You still have some time. You still have some time, Sid.
Okay, excellent. Okay, so this first section, I'm going to go over our ability to be able to guide over sequence-based properties without any additional training. So specifically, I'm going to talk about 4 specific tasks: enriching for rare amino acids, generating symmetric and repeat proteins, functional sequence scaffolding, and multi-state design. So how do we exactly bias for certain amino acids? Well, the process is actually quite simple based on our sequence representation. So we go ahead and noise, started with completely random noise, give that to the model. The model is going to output a denoise sequence. We go ahead and select the top K positions with the highest propensity for the amino acid we care about. So in this case, we care about tryptophan. We go ahead and literally add on weight to those specific logits. And then we go ahead and add on weight to those specific logits. And then we go ahead and task the model to denoise, and we iteratively do this process. And as time goes on, the model starts giving tryptophans, in this case, to those specific positions. Now, this paradigm actually works fairly well because we chose to do this on amino acids, which the model does not sample very much. If you remember from our previous slide, and these specific ones were valine, tryptophan, cysteine, histidine, and methionine. And so unconditionally, we see that the model is not sampling those amino acids very much at all. But with our biasing, we can get these amino acids to represent approximately 20% of the protein. And then to see whether this whether these proteins are occupying a unique place within sequence space, we do some multidimensional scaling, and we see that there's two distinct population. And then we actually went ahead and went to the lab and tested these designs and saw whether, one, do they elute at the specific desired volume with size exclusion chromatography, what tells us whether these proteins are monomeric or are they aggregating. And so in this case, we have SEC here, and we see that our models, our designs are eluting at the expected volume, telling us that they're monomeric. But another thing to note about tryptophan, it has specific spectropop, spectroscopic. Okay, I'm forgetting the specific word. It has specific properties that make it absorb quite strongly. And so we compare with a design that doesn't have a lot of tryptophans, we see that absorbance is quite high. Which is certainly cool to see, and it might be useful for imaging technologies in the future. But then the next study we did is something called circular dichroism. And what circular dichroism, it tells you the overall secondary structure content of our protein. And so if a protein is alpha helical, it will have a specific spectra, whereas if it's beta or alpha beta, again, would have a more distinct spectra. And so in this case, this is a spectra we'd expect for an alpha helical protein. So that was really nice to see. And then here we're upweighting cysteines, and cysteines are interesting in that they form disulfide bonds, but if the cysteines are not located in the correct place, they could end up causing the protein to aggregate. And here we see that our proteins, even though we're only upweighting for cysteine, but not defining where these disulfide bonds are made, we see that the proteins are actually able to elute at the proper volume, letting us know that they are not aggregating. And in addition to that, if we look at the CD, it's all, it has the expected alpha helical trace. And in addition, because these disulfide bonds being formed tend to make the proteins highly stable, if we increase the temperature to 95 C, we see that the CD spectra largely remain the same, indicating us that these proteins are stable even at high temperatures. And then to make sure that we are forming the actual disulfides, we go ahead and do mass spec in both reducing and non-reducing conditions. And from this, looking at the mass difference, we can see that the disulfide bonds are formed for most of our proteins, except for this last one, where only 4 out of the 5 expected disulfides were formed. And then for this one here, we're upweighting valine. And here, again, we see, again, we see that the protein is stable at high temperatures and also has the expected CD spectra with some beta content. Now, what to note about valine is that it increases the propensity of the amino acid has this property where it increases the propensity of beta sheets. And so without biasing, the model tends to adopt mostly helical proteins, but with valine biasings, we see we can actually change the structural attributes and we get more beta content. And again, this is showing the model has distinct understanding of the sequence to structure relationship. And then moving on from here, we did some repeat protein experiments. And how we do the repeat protein design within the model is actually quite simple. We have the model output an X naught. We take the first say XN residues, and then we repeat that however many times. So for instance, if we want a repeat protein with 5 repeat units, then we take that say N residue section and repeat it 5 times. And so this was our symmetrization procedure. It's quite simple, but we found it to be quite powerful. And so here's a repertoire of proteins we went ahead and synthesized, generated that are either mix alpha beta, all alpha, And here, what's cool to see is that, again, they elute at the expected volume, have the expected CD criteria, and are stable even at high melting temperatures. And we were actually able to solve a structure for one of these designs. And we actually see that we get quite high atomic accuracy. And here you can see, even at this resolution, the side chains are well in agreement with the designs with the generated proteins. And then in this section, we were curious whether we could actually scaffold functional sequences. So how we do this process is we task the model to scaffold a cleavage site. And so this cleavage site would be in the presence of a protease would get cleaved, but without the protease, it will not get cleaved. And so we scaffold this cleavage site. And the only specification we do for this cleavage site is we specify it has to be a loop. And the reason we have to specify it's a loop is because if it adopts a secondary structure, it'd be much harder for that protease to actually go in there and cleave. And the other thing we task the model of scaffolding is a active peptide. And here we just named it PepSeq. Now this PepSeq, we don't specify any structural constraints other than it needs to be masked, other than it just has to interact with the protein. Now, the peptide we use here has some specific properties. Now, whether that may be a barcode or lysis. And so for this specific experiment, sorry, for this specific experiment, the actual peptide is a lysing peptide. And so the task is, can we scaffold this peptide, which lyses, and specifically this peptide we're using is melittin. Can we scaffold it such that without the protease, it's masked, but with the protease, it's unmasked? And so the reason you want to do this is, say you have a patient with cancer and there is a specific tumor environment, and that tumor environment has an increased amount of a specific protease. In this case, the protease is furin. And so the question is, can we design a protein that has this logic in it where it exposes this toxic payload only within the presence of cancer cells? And then again, this is how the entire process is laid out to the model. You don't specify any sequence for the scaffolding region. And then you specify the sequence for both the cleavage site and the bioactive peptide site. And so in this case, we give the sequence for the furin cleavage sites and the sequence for the melittin toxic peptide. And then for the only secondary structure constraint we put on the model is only at this cleavage site here, which we specify we want it to be a loop. And then give all the information to the model, and the model gives us a sequence to structure pair that agrees with its conditioning information. And so we went ahead and synthesized this protein and then saw that it elutes at the expected volume and has the expected CD spectra. But next we want to see, in the presence of furin, does the peptide get released? And so furin is here, gets cleaves at the specific cleavage site, and then outputs are melittin. Now the question is this logic maintained? And when we went ahead and tested in the lab, we do in fact see that it is maintained in this conditional hemolysis experiment. To the left here is we have our controls. Triton is known to go ahead and lyse cells, and then melittin alone, we see that it lyses cells alone. But if we just leave the cells in buffer, no lysis. We just leave the cells with furin. Again, no lysis. And then the cool thing here is we leave the cell with the design, no lysis. But if we give the cell both design and furin, we see lysis. And so here, this is confirmation that we are able to conditionally lyse cells. And so this is a really cool application. And it was really, really exciting once we got this result. Okay. And then the next problem we sought out is actually one of the big problems within protein design, because within protein design, we've gotten really good at creating these rocks, these really stable rocks, but these rocks can't really adopt distinct states. Now the question is, we have this advanced model, can we actually apply it to this problem task. And so we conceptualize this problem where, say you have a parent protein that has an alpha beta fold, but let's say we go ahead and cleave it. Can we get this parent protein to give two distinct children that are all alpha? And so this was the problem statement, and we wanted to see if we could tackle it. And so we actually have a lot of tools with our tool. We have a lot of machinery within our toolbox. And the very first thing is we are able to condition on specific secondary structure content. And so we task the model with we condition the model with the specific DSSP string for the parent and the children. And so the parent, we task it with DSSP with both alpha and beta. And then for the children, we task it with just with just all alpha. And then and then I guess another so this is what we condition with, but one thing to note is there's actually some applications for this formulation of problem. And so in the case of, say, we want to just do fold switching, we do what we did here. But say you want to do binder design, say we want to do binder design, we can go ahead and have a binder with target adopt a specific state. So in this case, we want to do strand pairing. But say when we don't want the binder there, we can just make this all helical. And so now you have that conditional state. And then there's also some applications with sensor and hinges. Yeah, I guess we can discuss that later. But so with this DSSP conditioning, we give all of this to the model. And then we have the model output both the sequence and structures corresponding to all of this. We go ahead and take the outputted denoise sequences, we average them, add Gaussian noise, and then we go ahead and take this xt-1 and give it back. And through this iterative formulation, we're able to get a sequence that adopts all three distinct structural states. And then here are AF2 predictions of such a family of designs. We were actually able to solve the structure of the parent. And here we see really close agreement between the parent and between the parent predicted structure and parent NMR ensemble. Now, it was really difficult for us to get structures of the children, but we were able to look at the NMR spectra, and it actually does give us some information. And here we're looking for distinct differences between the environmental conditions of each residue. So if they're in a different environmental context, the given points for each residue should be different from that of the parent, and we do see distinct shifts between the children and the parent. But our collaborators were actually able to get a lot more information, because it seems that if you look at the actual width of these NMR spectra, it gives some understanding of the actual secondary structure content of the designs. And so things with increased beta sheet content would be towards the bottom left here, whereas things with increased alpha helical content will be towards the top right here. And so if we specifically look at this family here, we see that the parent, it's towards the left quadrant here, whereas the children are in the upper right quadrant. And we see this for all families we've tested, indicating to us that we are, in fact, have been able to design proteins that are able to fold switch depending on the environmental condition. So this was super exciting to see. And it was actually a lot of work. So happy that we're done with that. And then the last thing is we wanted to see if we could do this notion of iterative guidance or active learning. So that is, can we take experimental data and continuously collect this experiment's data in a design test cycle and see if we can guide with this data and see if we can get increased activity. So that is, say we have some design task, we generate a bunch of proteins, we test them experimentally, get a sequence activity data set, train a surrogate function or classifier on this data, use that to guide the generation of the next round, and see if we can iteratively increase the activity. As we did this experiment with this data set that's pretty exhaustive. And so we could actually mutate to every single possible mutation on these 4 sites and then actually verify on on that data set and see if we could actually increase. And we see that our model, ProteinGenerator, it's in purple here. We see with each successive round, on average, we're increasing the average activity. But in addition, if we look at the maximum activity, we also see that we're continuously increasing in just as few as 3 rounds with only 96 data points per given round. And so in general, in summary, ProteinGenerator allows you to do quite a bit. So for once, we can do sequence guidance. And here I've shown experimental data for enriching for rare amino acid, repeat protein design, and sequence motif scaffolding. In addition, we can also do concurrent sequence structure guidance, and we've done this with conditional peptide. I've shown you experiments for conditional peptide caging and multistate design. And I've also shown you some preliminary work in seeing we can actually guide for specific activities with sequence to activity data sets with the GP1 experiment I just went over. And so big thank you for all my co-authors and collaborators, and then my advisor for all the hard work we were able to get done to get this paper out. And so with that, are there any further questions?
Thank you, Sydney. This is really a wonderful talk. I don't know whether Ben has raised a hand. Ben, do you want to say something?
Yes, thank you very much, Lukman, for organizing the session. And personally, I also want to thank the presenter, but I just have a few questions. I may not have the background of the AI and the language models and all those things I've been talking about, but I actually want to know if one has a sequence for a peptide and they actually want to move backward to the specific DNA sequence for that peptide. Can this model guide that kind of transition? If you want to get the sequence, I mean the DNA sequence for a certain peptide, and you simply want to amplify in E. coli for bulk production, such a kind of.
So the model can't do that. We're specifically just only working within amino acid space, but there are newer models that are coming out that could do that. And I think George Church's lab has a specific model that can do that. So I would recommend just looking over their work and see if that would be useful for you.
It's called the George what?
George Church.
Okay, George Church. He's also a member of the University of Washington.
No, he's at Harvard or MIT. One of those.
George Church's work.
Yeah.
Okay. So I don't know whether Ben, they have answered your question.
Yes, thank you very much.
Okay. Personally, this is exciting, and as someone from software engineering or computer science, I would say diffusion models are becoming like powerful every day, and it looks like, I don't know, Sydney, you will guide me, but it looks like the space for them to do more wonderful work within biology is still wide. And probably there are still unexploited architectures that can handle diffusion process. With that, I want to thank you once again. Thank you for accepting to really share with us this wonderful work. We are honored. And I want to also